introduce Fred now. Professor Fred Coulon from the University of Cranfield, who's kindly organized today's session and is our chair, and he'll be assisted later by Tony, who I've mentioned, from Heriot-Watt. Right, over to you, Fred.
Thank you, Angie. Well, I had an easy job, actually. It's more Barbara and Zeugen that kindly accept our invitation. So, as Angie explained, we're on the EBNet, on Environmental Biotechnology Network, and we are having this webinar series to trying to stimulate the community around perhaps research need or barrier to overcome or new challenge and area upcoming, but also what's currently happening in the field. And both Barbara and Zeugen are leading researchers, especially on the water-based epidemiology, and also in the recent with the pandemic on COVID-19 and how the wastewater come to focus. So we are kindly inviting them to give us some insight about the ongoing work, and we will welcome questions, but also opportunities to develop perhaps some white paper or new research agenda, and how we could potentially create some new opportunities within the community in UK, but also beyond, because we have also overseas delegate today, which is great to see. So our first speaker is Barbara, and I will give her the floor where she will provide us an overview of the lead work done at Bath University on the water-based epidemiology and how we can use it for tracking down chemicals and understand the impact, environmental and health impact, and specifically with the wastewater. And then we please use the chat function if you have questions, and we might be able to allow a few questions before moving to Zeugen with the sensor-based paper research. Over to you, Barbara.
Thank you. Thank you for your introduction, and thanks for inviting me to this meeting. I'm going to switch camera off and upload my presentation. I hope it's going to work. So I hope you can see it now. I'm going to make it full screen and start.
You're
perfect. Good. Working. I'm going to use this nice pointer. Okay. So thanks again. I will start with a bit of a background of why we do what we do. I think we all recognize one big challenge, which is rapid urbanization and population growth that by definition will impact public environmental health. So there are more of us and we live in urban areas. This equals larger density of population, and this obviously equals the high likelihood of infectious disease spread, but also due to lifestyle choices can be linked with non-communicable diseases such as cardiovascular diseases. But I think we all agree with one very simple statement, and you see written in red that we have failed globally in terms of detection and management of SARS-CoV-2, and there are various different reasons why this has happened, and the reasons will be of national, local or global importance. But I think one of the key aspects that we have missed out on is having a system in place that can inform us of any new pathogens coming up with potential deadly consequences we can see with SARS-CoV-2. So there is a clear need to have an early warning system, in this case for public health, but also environmental health, diagnostics that operates on a large scale and in real time or near real time. This is a genuine importance. So our idea is to develop a public environmental health early warning system via a slightly unusual matrix or way, and this is via urban environment fingerprinting. And I would like to emphasize something very important. We do not want to replace with our tool any other tools that are already in place. We just add to the toolbox. This is yet another tool that can actually help us with triggering some interventions of localized scale. And I will introduce you to this approach that we have developed. And by us, I don't mean myself and researchers working with me. We have thriving, very strong networks, and key to success to this approach is that we started working with each other from very, very early on. And I will give you a bit of historical perspective, also in the context where we are with SARS-CoV-2. Although I'm a chemist, apologies, I will focus on chemicals. This is what I do, but I will also link with SARS-CoV-2 because we are involved in some of the projects. So, how does urban environment fingerprinting work? And especially urban environment. This means soil, water, and air. But we focus on wastewater, and I will now give you reasons why we do this. So. Every hour, minute, second of our life, we're exposed to various different agents. They can be physical, chemical, biological, so there can be some chemicals, or they can be pathogenic organisms, and they are foreign to our bodies, so we call them xenobiotic. And obviously, if they enter our bodies, the certain internal mechanisms will be triggered to remove them, and this is manifested with the formation of various different we call them markers, so chemicals, can be metabolites if metabolic processes are in place. We have pathogenic organisms excreted, and also we can have changes at genetic levels. We have certain proteins being excreted, for example, CRP, C-active protein, as a result of inflammation in our body. And all these markers, or biomarkers, we call them, biomarkers of effects, as opposed to, so these are the ones here, as opposed to markers of exposure, so actual stresses, xenobiotics that we are exposed to. They end up in the sewage system, and obviously those that end up in the sewage system will have certain properties. So they are soluble in liquids, they are excreted with feces or urine. So when looking at wastewater, we're focusing on those that are excreted with feces and urine. So mainly we actually focus on metabolites of chemicals, but not only. And once in sewage system, they will end up in wastewater treatment plants. So I'm sure most of you will be aware that wastewater treatment plants serve well-defined communities. So from 1,000 people to a few million people. And water utilities have a good understanding of where they collect wastewater from. So if we collect a wastewater sample and measure various different markers of exposure and effect, we can provide a fingerprint of communities' health and lifestyle. There are various different things that need to be looked at and considered, and I'm going to focus on those because they're extremely important in understanding of what WBE can do. So conceptually, it's a very simple approach. As you can see, someone might say, Barbara, what's a big deal? You just go out there, collect wastewater sample and analyze it for various different things. And actually, methodologically, it's highly, highly sophisticated. We are really looking for a needle in a haystack. And we do what human biomonitoring researchers do, but we work with much more complex matrix. So not urine, it's a highly diluted matrix with various different potential interferences to our analytical signal. So it's a bit of a challenge in there, but with many compounds, we have managed to overcome it. And also, in order to provide quantitative measurement, we are after quantitative measurement. We want to know how many people are exposed to various different things. We need to have an understanding of the wastewater sewage catchment system. And with the new wave of research, I think if you might not have heard about WBE last year, I'm sure you have heard about waste-based epidemiology this year. It's also called wastewater surveillance because of SARS-CoV-2. SARS-CoV-2 gets excreted with feces into wastewater, and we can look for its RNA in wastewater. But the issue that's when I see the developments in the context of SARS-CoV-2 is that people measure for RNA and not necessarily take into account all the complexities of the sewage system that need to be taken into account to provide fully quantitative measurement. So I will give you some examples on how we started this in terms of some of the chemistry work. But before I do it, I use this slide just to make a link with what is done at the moment in terms of public health, so the kind of human biomonitoring dimension. So in human biomonitoring, urine samples from thousands of people would have to be tested to verify community-wide health. And this is done, but it's expensive, and we cannot really take into account whole community. but only one waste sample will do exactly the same to assess communities' health. And what it does is that it includes everyone. So we don't necessarily have to collect samples from individuals. We have a pooled sample. There is a limitation of this as a result. We don't identify individuals, so we cannot really introduce an interventions needed at the individual level. And I've mentioned this earlier on. Our tool should be used more as an early warning system. So if we do large-scale monitoring globally or nationwide, and we can identify areas that require more focused interventions, this is the tool that is going to deliver on this. While we always have to have the traditional human biomonitoring epidemiological studies when going into communities, because this tool will be useless there simply because we don't know who is ill and whether it's male or female, or whether this person has some underlying health conditions. We cannot deliver on this. So just to summarize, we really are delivering on the more kind of generic tool that's more like an early warning system that can help with identification of issues at the community level. And I always also show this slide just to show you where we started. And the idea actually came out from environmental scientist Christian Doughton. Christian is retired now. He used to work for US Environment Protection Agency. And if some of you are involved in pharmaceutical work, he was a really well-known figure in this area. And while doing all the measurements and looking at various different results, he came into conclusion that if we see, for example, metabolites of illicit drugs, they must have been consumed. If they're consumed and we see this, we might be able to calculate how many people consumed illicit drugs in the community. And this is how we all started, really. So by 2010, we had a European network. We called ourselves SCORE Group, which is Sewage Core Group Europe, not the fanciest name, but it is with us even today. And when we started working together, we attracted a large funding from European Commission and now also from UKRI. So in black, we have huge support from UKRI. But I want to emphasize that we attracted this funding because we work together across Europe. So I think your network, I think you are doing exactly the same, teaming up with others and delivering on some of the big challenges. So, and I mentioned, oh yes, and we started with illicit drugs and alcohol, also nicotine usage or consumption via waste-based epidemiology, and then we moved on into food toxicants and infectious disease and also non-communicable diseases. And as we started with illicit drugs, I will give you an example of cocaine. And how our approach works and what needs to be looked at when extrapolating to SARS-CoV-2. Again, you can't compare SARS-CoV-2 with cocaine, and it's a chemical, biological agent, but there are certain things that we need to be looking at, and they're not looked at by many in this field at the moment, focusing on SARS-CoV-2, which might actually, unfortunately, skew the results. So what do we look out for when trying to calculate or estimate cocaine consumption across the community? We won't be looking at cocaine. We will be looking at its metabolites, so indigenously formed chemicals that are linked with exposure. And luckily for us, cocaine metabolizes really well. And its metabolism actually, metabolites are linked with the route of administration. So smoking will lead to this metabolite, alcohol, co-administration with alcohol will lead to actually toxic metabolite. We tend to focus on the main metabolite, benzoylecgonine, produced by cocaine. Produced by metabolism, and with SARS-CoV-2 we don't have this situation. We are focusing on RNA, and as a result, it's difficult to distinguish between SARS-CoV-2 that found its way into the sewage system, for example, via runoff, and the one that has gone through human bodies. So there is a huge limitation here, and might be affecting a lot the results that we get. So what else do we need to look at? So metabolism is critical, and we need to have an understanding of how much of a certain chemicals gets excreted as a result of consumption of certain quantity of a drug. Now, very critical and still very important is population size. We need to know how many people contribute to our sample. And we tend to use population equivalents provided by water utilities. And we try to also calculate our own. This is one of the key limitations still. So if you have any ideas about some really good marker that is linked with humans and nothing else that might be linked with the environment, please let us know, because it will save a lot of time, make our life easier. So population size is very important, and especially in areas where we don't have very well established sewage infrastructure, so in lower resource settings. Now we need to also have an understanding of flow of wastewater, because we need to move away from concentrations into daily loads. And you know that for example, in the UK, the levels of either the virus or chemical will be hugely affected by rainfall. So we need to take this away from our equations. And also we need to know the biomarker that we're looking at is stable in wastewater, so not subject to some microbial processes in biofilms in the pipes, similar. But once we know this all, we then can plot our graphs, and this is actually the data that we produce as core group. So the Sewage Core Group Europe, our data feed into European Monitoring Center for Drugs and Drug Addiction, and this is actually their database, and they can see changes in terms of drug usage across Europe. If you are interested, please have a look at this link. Now I will move on to what we do in our group. We do focus on the calculation of illicit drug usage, but we do much more. And here there are just few examples. We work across environmental, public health arena, and non-communicable and communicable disease. But key to success is really to know what to look for, so what kind of biomarkers to focus on, and understanding the stability, the performance, sensitivity of methods. We need to develop methods. So this is most of the time we actually spend in our labs developing methods. And what are we doing at the moment? Starting with endocrine disruption, we're looking at some of the industrial chemicals, trying to calculate, and I'll give you an example of bisphenol A, try to back calculate our exposure. We do a lot of work with antibiotics and AMR. There is this key issue in terms of linking environmental AMR with public health dimension. We also develop methods for biomarkers of effects, so for example, oxidative stress, inflammation, and infectious disease. So we do work with SARS-CoV-2, although, as I said, this is work in progress, and I'm not going to show results today. And our work can feed into various different areas. So from evaluation of interventions through to exposure studies, spread of disease, early warning system. Actually, we are developing a warning system in Africa, and I will give you just very brief snapshot of this. So BPA, bisphenol A. Bisphenol A is one example, really, a chemical that we work with, and it's you must have heard about bisphenol A. It's an endocrine disruptor, and it's used in plastics. And it's been banned by European Commission from, for example, children's toys and bottles. And European Commission estimates the main exposure to BPA is via food, because BPA is in plastic that is used for food packaging. So if you are a fan of fast food, microwaving your food, you will likely have high exposure to BPA. So what we try to do, we try to look at wastewater-based epidemiology and whether we can back calculate exposure to BPA. There is an issue with BPA and other chemicals that are not intended for human consumption. We have very limited understanding of metabolism, if any. So we did actually study across 5 different cities in southwest UK. So these are 5 cities, towns, served by 5 different wastewater treatment plants, as you can see here. 3 of them, those in grey, have only communal contributions, so there is no industry in there. And those 2 in orange have industrial presence. Up to 30% of industry. That's mainly food packaging, food. There's also paint formulation. So industries that are very much linked with BPA, unfortunately. And these are the results that we get from wastewater analysis of BPA only. So you can see 5 different treatment works, and one of them has higher levels. And these are population normalized daily load. So we have normalization of population size and normalization to flows of wastewater. So these are the two key variables that we are dealing with in wastewater-based epidemiology. So you can see that in wastewater treatment plant E, with population equivalent of almost 1 million, we have this spike. And the question is this really, does this spike indicate internal exposure, or is it basically direct dumping into wastewater by one of the industrial sites? And we can't really tell because we are looking at parent compounds. So the key to success, and I'm again wanting to link with SARS-CoV-2, is to look for more than the stressor itself. And so with BPA, we need to look for some metabolic biomarkers. And unfortunately, this is not a well-known knowledge. BPA is not intended for human consumption, so we don't have information about this. So in our group, we developed a tool that will allow us to identify biomarkers of exposure, so metabolites. And this is the kind of stepwise approach. I'm not going to get into details. I'll just briefly skim through three slides just to give you an idea about complexities of what we are dealing with. So in step one is a biomarker discovery pipeline. Once we know what kind of biomarker we are to look for, then we move on into data mining, and then we deal with large data sets in terms of back calculation of community-wide exposure. So very briefly, biomarker discovery, if there is no information available in literature, public domain, we need to combine kind of human in vitro experiments, so human liver microsomes, S9 fraction, and we combine them with pooled urine assays and wastewater fingerprinting assays, and we compare and contrast the data and try to draw conclusions about which metabolites are the ones forward. So we do modeling to start with to see what kind of metabolites can be formed, and we have 3 here, but only one follows the following. So it's human derived, is stable in wastewater, so microbes are not too fond of it, and is also quantifiable in wastewater, so we have methods that can deliver on it. So out of the 3, 1 is a possibility. And once we know what kind of marker we are looking for, then we dig into our high resolution mass spectra. We have databases set up. And again, I'm not going to get into details here, but just to show you complexity of this. But we have tools developed to achieve this. And once we have this, then fun starts in terms of looking at what's happening at the community level. So we have the same 5 sites, and we're actually covering a large area. So in the southwest UK, so over 100 kilometers wide, 5 cities covering 75%. Over 75% of contribution from people in this particular catchment. So a good data set to work with. And we have results here for 5 different cities. And these are population normalized daily loads, and this is BPA sulfate. So we're not looking at BPA itself, we are looking at the marker. And this is sampling over 1 whole week, so Wednesday to Tuesday, including weekend. So we can see some really interesting things in these 5 cities. So one interesting is that there is the weekend effect. So weekdays, high levels of exposure, weekends, lower level of exposure. You might not see this very well in the low levels full stop, but there is still this effect. It is very much visible in city B and E with much higher exposure community overall. So this is an interesting one, and there are 2 reasons for it that we can come, and this is our wishful thinking. One is maybe we have a bit healthier lifestyle and we don't eat fast food during weekends, or more likely we don't have, in sites that have industrial presence, we don't have occupational exposure. So these are these 3 towns and cities that don't have any industrial input, and you can see overall levels of our exposure to BPA is low, which is very good, but we have very high levels here during weekdays in city B and E, and these are the 2 cities that have high industrial presence that is linked with bisphenol A usage, so we have clear occupational exposure here, which wouldn't be recognized, would get unnoticed if we're doing traditional biomonitoring studies targeting whole population, not necessarily workers. Now, I mentioned earlier on that we need to look for metabolites rather than parent compounds, and this also links with SARS-CoV-2 again. I don't have answers in how to deal with SARS-CoV-2 at this stage. And there is a very simple example here for carbamazepine. Carbamazepine is a pharmaceutical is prescribed for as an antiepileptic drug, and it is prescribed at stable levels across communities. And here we have carbamazepine and its load and also intake, so on both sides, and its main metabolites, carbamazepine 10-11 epoxide. So if we focus on parent compound, you can see what happens here. On Sunday, we have massive spike. And if we were to look and calculate exposure based on carbamazepine intake parent compound, we will be hugely overestimating this. Why has this happened? This is direct disposal of a few hundred pills down the drain by possibly one household. And why do we know this? Because when we look at metabolite, we don't see the spike. The community still consume the same quantity. It's just someone somewhere dumped huge quantity of carbamazepine. This actually has a huge ecotoxicological acute localized consequences. But again, it will get unnoticed by regulators because we don't test at this resolution level. And I wanted to also, if we pay attention to details, we understand the catchment very, very well, we can link, end up with having very, very good results in terms of carbamazepine intake. So the red line here, so the columns indicate loads, so milligrams per day per 1,000 people, while the red line indicates carbamazepine intake in milligrams per day per 1,000 people after taking account of the data. Okay, so this is our estimation. And this is our postcode prescription of carbamazepine. So we kind of compared top-down, bottom-up approaches. So we calculated carbamazepine presence in wastewater based on prescription data. And we also calculated prescription carbamazepine intake based on wastewater data, and we landed here. So we have very well-defined catchment in where we are working, and it can be used for system calibration. So yes, we have done a lot of work in this area with and still working. It's a large data set with 300 compounds, and they are mentioned here what we are looking at the moment. And this is just an example of what we can do via chemical mining of wastewater. So this is fluoxetine, so known as Prozac, pharmaceutical that we prescribed over longer term. We wouldn't have this weekday versus weekend variability. So you can see stable consumption across. So on this side, we have again daily loads of biomarkers. So these are all metabolites. No fluoxetine is metabolite of fluoxetine. Benzoylecgonine is metabolite of cocaine. BPA sulfate is metabolite of BPA, and 3-PBA, a very interesting one, is a metabolite for pyrethroid pesticides. So what can we see here? That fluoxetine intake, so fluoxetine intake will be this red line here, is stable throughout the week. Cocaine intake will be higher during weekend, and we know that it's a club drug, so we have weekday increase, and it goes alongside decrease in weekend uptake of BPA because of no occupation exposure. Interestingly enough, we have this weekday spike of pyrethroid pesticides, and we've been thinking about why this is happening. Maybe because we have healthier lifestyles and we tend to consume more vegetables. As a result, we expose ourselves to pesticides. So you can see what this tool can provide us with. Very important information about exposure studies, high resolution information about exposure studies. And again, I want to emphasize, this is per 1,000 people. We just don't know what the individuals do. We don't want to know. We want to know what's happening at the community level. And yes, just very briefly, that the results that we get, I'm not going to bore you with details here, but you can see that some of the non-communicable disease pharmaceuticals are stable uptake, 5 different cities. So across the catchment, we do the same stuff. While when we move to illicit drugs, this will be city dependent. Smaller cities, lower consumption, larger cities, large urban areas, higher consumption, and this is per 1,000 people. More industrialized areas will have higher exposure to pesticides and some industrial chemicals, while more residential areas will have low exposure. Now, I'm not sure how much I've already taken in terms of time. Now, just wanted to briefly let you know about AMR, but there is a huge problem, an issue that we have in terms of linking antibiotic usage and its discharge into the environment, the discharge environment, and whether we can link this with AMR, environmental AMR. So we did some studies using WBE to understand this a bit better. So this is our European work, part of SUPROF project, and just a very quick result. We studied a few different cities, and here we have results only for fluoroquinolones as of synthetic antibiotics. And we have also results for QNR genes, so the resistance gene that is linked with the fluoroquinolones. So we have 7 cities. Bristol is here. We have cities across Europe. And you can see when we look at the gene across Europe, levels are stable. And this is concentration, so number of copies per liter. Now, with antibiotics, we have this really nice trend of higher levels in Milan and Castillon, and this is in nanograms per liter. And one might want to look at this result and say, oh, definitely there is no correlation. There is really no correlation between antibiotics as genes. And the reason why there is no correlation is because this data is not normalized. And we've done a lot of work, as you already know, in knowing how to normalize data that is fit for purpose. So when we compare, for example, Castillon and Bristol, Castillon has much higher concentration of antibiotics when compared to Bristol. But is this really higher usage? In Castillon, it rains less. In Bristol, it rains more. So in real terms, the situation might actually be reversed if we normalize to flows. So this is how normalization to flows looks like. When we normalize to flows, you can see Castillon suddenly does not have higher usage of antibiotics. It just had very high concentration of antibiotics in wastewater due to no rainfall during sampling. But what we can see, simple normalization to flows, and we start seeing patterns. So these are now loads. So QNRS gene, number of copies per day, and this is antibiotic milligrams per day. What can we see here? There is a correlation between the prevalence of antibiotics and genes. So if you can see Milan stands out now, it has not. When you look at concentration, there was no correlation at all. Here we have clear correlation. The higher the usage of antibiotics, the higher the presence of resistance genes. And Castillon, as you can see, is not looking as prominent as it would be when looking at concentration levels. We have a bit of a problem with where Oslo and Bristol is, because we know that in Northern Europe, especially in Oslo, we have in Norway, there is much lower usage of antibiotics. We know that the usage of antibiotics is much higher in Southern Europe, so hence the link with resistance. We cannot really explain this higher levels of QNR gene here. Still, pattern is clear. In Netherlands, very low usage of antibiotics, very low presence of the resistance gene. And what is also important here, if you look at population size served by treatment works, there is a clear correlation of people that provided the sample. Okay, so we as individuals are, according to our data, responsible for the resistance in the environment. So why do we think so? When we then normalize the population size, you can see this results plateau. So individuals, number of people contributing to the sample will be proportionally, there will be proportional correlation with resistance genes. So here we have over 1 million people, as opposed to 180,000 in Castillon. So population urbanization are key drivers of antibiotic resistance genes in the environment. And we've done the same in our UK study. So again, the same 5 sites, and we looked at 5 different treatment works, and no surprise, site E, that serves almost 1 million people, has the highest daily load of antibiotics and resistance genes. So wastewater data can provide us with very, very important information. I'm going to leave it there for now, just give you a very quick overview of the projects that are ongoing. So more data is being produced and dealt with. We actually run into serious issues with managing large data sets. So we are looking at global exposure to chemicals in consumer products via wastewater-based epidemiology, and this project is led by Stuart Harrod, based in Birmingham. It's European Union funded project. It's been running for a few years now. And we have also a few GCRF projects when we look at, in this case, in Stellenbosch, look at correlations between antibiotic resistance genes, so linking on with the work we've already done and trying to develop an early warning system at the city level, a focus on spread of disease and AMR. And I'm just only going to let you know that we have these two projects, but no data. Data is being produced at the moment, linking SARS-CoV-2 and other also non-communicable diseases with building early warning systems in Africa. So just big thanks to everyone who contributed. I'm the one presenting today, but this is the work of many people, and we had a huge support from European Commission research councils, but also I would like to emphasize the input from our best collaborator and also funder of our work, which is Wessex Water, so our local water utility in the UK. Thank you very much.
Thank you very much, Barbara. Very comprehensive. It was maybe one or two questions. It was a question about whether it's a rule of thumb for what contaminant you can detect and can't detect in wastewater, or is always a challenge. Or do you know in advance what you might be able to detect and what you can't detect?
What I can detect, what I can't detect. Okay, I'm going to stop sharing.
Yeah.
Yes. It's down to what kind of tools we have, really, and how fixated we are on the idea of detecting something. So I believe that in many markers we can detect. It's just whether we have enough time and resource to make this happen. So what we tend to do in our work, because we do multi-residue measurement, it takes time and a huge resource to analyze, collect samples and process. So we do test, for example, 100 chemicals at a time. And to be honest, now we're actually moving away from target analysis into non-target, so we then retrospectively analyze for chemicals. And hence we use multi-residue methods that are not perfect. So they're good for some compounds and they're not good for others. And sadly, this is what we have to deal with. But if there is a chemical that we really are after, and it's very important, we can modify our methods that they are fit for purpose. But in terms of wastewater, there is a very simple situation here. So all chemicals that are soluble in wastewater or they're not volatile will end up there. So they can be either in liquid phase or they can soak to particulate matter in wastewater. So we would not be looking for volatile compounds. They will be present in the air, and we'll need different methods for this and different approaches.
And you presented quite a lot on the population and daily. Is it mainly on passive sampling, or do you have like an online monitoring device allowing you to get more real-time monitoring, and perhaps thinking with the COVID, for example, we're trying to find early warning systems. So there's been quite a lot of publication trying to illustrate with down protocol how maybe we could save or get or earn days in advance before the peak. So a lot of the data obviously is trying to identify all this chemical, but is there any progress on online monitoring, or where do you see the needs?
Yeah, no, there is definitely a need, and Zügen obviously will tell you about this. And Zügen also was part, I'm not sure, Zügen, whether you're going to mention that you were part of SUPROF a few years ago, and you developed, I think, the first sensor, cocaine sensor, for real-time monitoring. We are not there yet. with chemical analysis, we do analyze in labs. We don't have sensors that can deliver on chemistry analysis. We tried, and yes, Zugen succeeded, but on the 1-2 compounds, we still believe that mass spectrometry can provide us with measures possible. That's how to deal with time lag. So we don't get results in real time. So we use this near real time, which it's down to interpretation what this near means. But again, it's down to results, because we could have a result in one day if we have people that can work day and night to deliver. It takes us longer than one day, yeah. which doesn't go together with the idea of early warning system. There are sensors that we are looking into, commercially available, that they can provide information about BOD, so kind of the proxies for environmental burden, but not necessarily chemicals. So this is, if we have people present today that do work within the domain of sensors, yeah, sensors for chemicals would be really brilliant, amazing. But I know that there are problems with sensitivity in the and matrix effects.
Yeah. So we got a good transition to introduce Zugen talk. Thank you very much, Barbara. And there's a few more questions Tony will share later on after Zugen.